Je présente rapidement Anna, qui est une amie qui a passé un an de sabbatique à Créteil pour initier toutes les recherches dont elle va vous parler et qu'on fait entre eux, donc les centres experts et Toronto. Elle est PhD pharmacienne. Elle a fait ses études de biochimie au Brésil avant de partir au Canada, où elle a été à Vancouver, puis à Toronto, où elle est maintenant professeure dans le département de pharmacologie et psychiatrie de l'Université de Toronto. Et elle a créé et elle est le directeur scientifique du Mitochondria Innovative Initiative qui fait des choses absolument formidables pour améliorer la compréhension du rôle des mitochondries dans les maladies mentales, mais peut-être qu'elle vous en parlera aussi dans plein d'autres situations pathologiques où on voit à quel point ces petits organelles intracellulaires ont un rôle absolument clé et qu'on connaît aujourd'hui de mieux en mieux. Donc elle va nous raconter des choses passionnantes parce qu'en plus elle est très pédagogue et que je suis sûre qu'elle va nous expliquer très clairement c'est ce métabolisme qui n'est pas si simple que ça à comprendre, mais vous allez voir que dans une heure, vous saurez tout. Anna, merci beaucoup.
Said I will be talking about how mitochondrial abnormalities might be actually important for bipolar disorder, and I hope by the end of this talk we all can agree that energy is life. Without mitochondria, there is no life, so that would not be different for patients with bipolar disorder or any psychiatric disease. If it changes in the mitochondria, will be affecting many different body functions that are associated with these diseases. Before I dive in on what I will be talking today, which is the first part of what my lab does, I thought to give you a little bit of an overview what I do and who I am. So I'm really as a mitochondria biologist and geneticist that try to understand mitochondria function in many different conditions, from health to disease. One of the biggest part of the understanding for us is actually understanding how mitochondrial function is involved on the pathogenesis of neurological and psychiatric disorder, with a particular focus on bipolar disorder. But I also study organ transplant and mitochondrial transplantation. My lab has been developing techniques that can be used and implemented in the real clinics to help regenerate organs. To give an example of a clinical application of this, working with the surgeons for a lung transplant, we asked a very simple question. How can we make a lung that has been put it into donation to actually be a better and healthy lung? Once we start asking that question, the first thing we notice that the organs were kept at 4 degrees. And 4 degrees actually puts mitochondria in a quiescent mode, meaning they are dormant. They are there, they are alive, but they are dormant. So we said, what about if we actually preserve the organ at 10 degrees instead 4 degrees? So the shock between 4 to 37 degrees is not that big of a leap. And at 10 degrees, mitochondria is still breathing. So we did that. We started with pig models, and we identified that actually maintaining the organ at 10 degrees actually made the mitochondria breathing better, having less inflammation in this organ, and lead to less ischemia damage in the organ, leading to a much better outcome when we did the transplant of the organ in the pigs. So we moved that to a human clinical trials, and now is actually clinical practice here in Toronto and is starting being implemented around the world. That led us to actually ask the question, can we even make organs better? Can we regenerate organs? So we start thinking, can we transplant whole mitochondria, healthy whole mitochondria inside organs, and ask if that mitochondria can actually be regenerating the organ that was damaged and was defined that to be no good for organ transplant at all. So that's the other part of my lab that we've been studying is mitochondria transplantation, where we use healthy mitochondria to transplant inside organs that are damaged to recover them. And so far, what we've seen is actually quite possible to do it for lungs and the liver. We are now moving into a study for kidneys and heart transplant. So as you can see, My lab is very cross-cutting, interdisciplinary, and I invite you all to visit the Mitochondria Innovation Initiative. It's a place where you can find a lot of information. You can find webinars that are free for everyone. We train students on understanding better how to measure mitochondria, how to access knowledge. We train clinicians to understand how they can diagnose mitochondria dysfunction in patients. So there is a lot, and they're all free tools that are available for everyone around the world. So let's dive in now, mitochondria and brain. So what you see here is a synapses. And when you look at that, you have remobilization of the neurotransmitters. You have to create these neurotransmitters. You have to exocytize the neurotransmitter. You have to endocytose that. All of that has to happen in the presynaptic neuron to actually take it all the energy and release the neurotransmitters here demonstrated as a glutamate going into the postsynaptic neurons. But mitochondria produces the ATP, adenosine triphosphate, that is essential, as you can see by all these arrows, for all this process that encapsulate your neurotransmitters and release them to the synaptic fence to ensure that neurotransmission is possible. So then you can start asking this question, well, if this process is not working very well, if mitochondria is not trafficking to the end, to the synaptic terminal, and you don't have enough and proper energy, perhaps the synapses will not be working very well. But then you can ask the flip question. What about if I have far too much energy and I start doing this process over and over again and releasing far too much? So now it's kind of easy for us to put it like a connection between energy and neurotransmission when you look at this as a necessity, chemical necessity of the neurons to have a balanced process to ensure proper neurotransmission that is not over hyperexcited or not down excited as well, but is right in the middle and properly function everywhere in your brain. This process is not only involved and is not only influenced, by the mitochondria in the brain cells, but from what is happening in your body. So changes in your metabolism in your body are affecting everything that is happening in your brain because mitochondria are sensors. They sense the environment around you. They sense the blood flow. They sense the glucose content. They sense the lactate content. They sense everything that is happening. And if everything is not working the way they like, they're going to try to adjust. Because mitochondria has one single goal. Keep you alive. Keep your heart beating. Keep you seeing. Keep you breathing. Keep you thinking. And they're going to do everything they can to actually ensure that you are alive. But when you have a metabolism in your whole body that is dysfunctional, that is not working properly, the mitochondria are going to do the essential functions on your brain. It will probably try to keep the basics going, or it's actually going to try to hyperexcite itself and try to compensate with the production of energy. And if that is happening, neurotransmission is going to be disturbed. A good analogy is like think about mitochondria as your bank account. If you have a good and healthy bank account with a good salary, good money coming in, you're going to go on and you're going to splurge on a nice walk on Champs-Élysées and buy yourself something super expensive and you like it because you had a great balance in your bank account. But if your bank account is like draining and you're not great today, your kids are asking for too much things, you have to buy a lot of things, you're going to do just essential. You're going to buy the food and pay the rent or mortgage of your home. Mitochondria does exactly the same thing in our body. If mitochondria is healthy, metabolism is healthy, sensitivity is healthy, you're going to have a sharp brain. You're not going to have brain fog. But if you don't have a healthy mitochondria, you're going to start having muscle pain, your heart is going to beat slow, your thought's going to be slow, because mitochondria is just trying to keep you alive at that moment because you don't have a healthy environment for it. So with that thought in mind, that's what we create this pathway that we study that is beyond mood regulation, is actually try to understand how this cycle between metabolic syndrome, cardiovascular disease, insulin resistance, and diabetes that is highly prevalent in patients with treatment-resistant depression, bipolar disorder, or depression. In fact, the majority of the patients are affected by these conditions. So we start asking ourselves, is this a cycle? What is cause? What is leading to? We needed to identify the biological pathways that increase vulnerability to a metabolic shift in these patients. Can we hack these pathways and bring back to a metabolic homeostasis? And if we don't treat this pathway well, if we don't pay attention that in our patients, we're going to lead to inadequate and ineffective treatments. We're going to escalate patient suffering and healthcare costs. With that note, I keep your eyes now to my right side in the slide, where you're going to see throughout the presentation quotes from patients with lived experience, how they see this metabolic pathways alterations affecting their own lives. So the first question we asked ourselves is like, is this a prevalence that we can identify? How many people that have mitochondrial disease, the classical mitochondrial disease, the rare ones, when the mitochondrial DNA is mutated and it leads to severe outcomes, where you're going to see from death to muscle weakness, to cognitive impairment, to strokes, to absolutely terrifying diseases for these families. So we went back into the Canadian records and asked how many people are living currently in Canada with mitochondrial rare disease. And is that a possibility that we can identify how many patients with the mitochondrial rare disease have also sought clinical treatment for mental health? And that answer is striking for me. 1 in 5 patients with mitochondrial disease also have a mental health condition currently living in Canada. And more importantly, look at the cost that added to the health care for these patients with the presence of mental health and mitochondrial disease. So I think that's a piece of evidence that doesn't give us a clue on how is actually being treated, what to do, but it gives a clue the importance of the connection between mitochondria and mental health. This paper actually is from a colleague, Alessandro Colasanti, in the UK, where he asked exactly the question, but now in the UK cohort. What he took was patients with lifetime of bipolar type 1 or type 2 and asked how many of the percentage of positive cases was for mitochondrial disease. In his case, was Leigh syndrome disease, the most common disease. And as you can see, actually, in patients with primary mitochondrial disease, was actually around 80% of the cases with this specific mutation. But now look at this, is bipolar disorder. The mutation that leads to Leigh syndrome is highly prevalent in the patients with bipolar type 1 more than bipolar type 2. Again, another piece of evidence linking mitochondrial rare diseases with bipolar disorder. The Wellcome Center for Mitochondrial Research in the UK also tried to inquire about this in their database. They didn't have enough information in their database to ask about the specific psychiatric disease, but they had something called psychiatric disturbance, where they ask for the patients whether their psychiatric systems are known, mild or transient, moderate or wandering specialist treatment, severe or institutionalized, or suicide attempt. And as you can see, the psychiatry disturbance grows up with the disease burden in these patients, highlighting the same thing. The more that you have psychiatric symptoms in patients with mitochondrial rare disease is a burden for these patients that needs to be cared for. From the evidences in the literature, I put a little bit of a QR code here that will lead to all the papers that is going to be shown in these slides. So please feel free to scan and get into in the QR code. So the mitochondrial DNA so actually let me pause here for a second. Perhaps it's better if I explain to you how mitochondrial genetics works. So mitochondria is a bigenome. What that means is mitochondria has its own DNA that is passed from your mother to every child. And the nuclear encoded mitochondria genes comes from your mom and dad. The mitochondrial DNA in the mitochondria forms about 14 proteins. The other proteins that forms the entire mitochondria, oxidative phosphorylation, the translation of mitochondria, come from nuclear encoded genes, and they are transported inside the mitochondria to form the mitochondria that you currently have in the body. So it's very important we understand that when we talk about mitochondrial dysfunction, we're talking about either mitochondrial DNA changings or nuclear encoded mitochondrial DNA. So in mitochondrial DNA, we can ask questions about, is this looped DNA called the mitochondrial DNA is decreased, increased in the patients with bipolar disorder? What we saw is a decrease on the amount of the copies of this mitochondrial DNA. But we also see an increase cell-free circulating mitochondrial DNA. What that means is that when the mitochondria gets stressed, it open up, release some portions of this mitochondrial DNA, and you can detect in the serum of your patients or plasma of your patients. We also see that the patients with the bipolar disorder, actually. This graph that you see here was my first paper that I published in this era. I'm not going to tell how long ago because it was kind of embarrassing. But this paper showed for the first time, actually, that patients with bipolar disorder had nuclear DNA oxidation higher than group controls. And yes, this was controlled by medication and by food intake, by diseases that they had in the time. And it was a paper that a long time ago was very difficult to publish because there was no one studying this area. So it was very little belief. And it's wonderful to see where we came in mitochondrial metabolism in psychiatry and how much we are now studying this as one of the main causations of main consequences of bipolar disorder. So in gene expression, that's the reason actually I moved to Canada. Professor Trevor Young published a paper with microarray studies in postmortem brain, where he actually demonstrated downregulation on several key genes on complex 1 to 2 to 3 to 4 and 5. And the ones that for me was really interesting was the downregulation on the NGF7 and NGFS8, because these two subunits are key subunits on the complex 1. Think like the complex 1 as an inverted L shape. It takes the electrons of your oxygen and try to pass them through an electron transport chain, an electron chain in your complex 1, and it gets to the middle and has to be released to go to the complex 2, like a cargo chain. And then this, if you don't take this electron out of complex 1, it's going to go back up and going to form reactive oxygen species called the oxidative stress. So this NGF7 being downregulated and a gene expression level was kind of interesting for me, because if you disrupt this first part of the oxidative phosphorylation chain, everything else will be disrupted as we go forward. So when we see, I asked Trevor, can I come and actually join your lab? I would love to do actually now to see if the protein levels of the NGF7 are changed in postmortem brain of patients with bipolar disorder and schizophrenia. And what we found was kind of interesting. We found that patients with bipolar disorder had downregulated levels of this NGF7 protein in patients with bipolar disorder on the BA10, which is a frontal cortex area. And we didn't find that actually from the same area in patients with schizophrenia. So that actually, for me, was a place where I said, I think we need to investigate more what's going on with mitochondria in these patient populations. Then we started evaluating a little bit more the mitochondrial metabolites. So when you take your glucose, you have to break it down until you get to a product called pyruvate that can enter the mitochondria and go into this citric acid cycle or TCA cycle that can actually now produce all the energy that you need to power your oxidative phosphorylation and form the adenosine triphosphate. So you can measure metabolites around this pathway that are breakdown, including pyruvate, or an essential one called lactate, Because if pyruvate now forms lactate, it can't go through the good cycle that forms 36 molecule, but it goes through a cycle that is actually fermentation is less efficient on producing ATP. It's not that it's not going to produce, it's just less efficient. So when we start looking into lactate levels in patients with bipolar disorder in the periphery, we actually see this is actually increased in these patients. And this is one of the most replicated finding in patients with bipolar disorder across the world. And also studies from Dostum Gur and many others looking into brain lactate levels found exactly the same thing to be actually increased in these patients with bipolar disorder. Yet we don't have a single paper, and I hope you are the group that's gonna do that, that's gonna measure brain lactate with MRS and peripheral lactated within the same patient population. We yet don't have the study being published. So with that motivated me to actually connect it with Marion, because the samples that you all have actually collected over the years is an incredible resource for the scientific community. I want to thank you, each of you, for put so much effort to clinically phenotype your patients so deeply, so beautifully, and to take the time to collect blood samples of these patients, because allow me as a scientist to now inquire how these changes in mitochondrial metabolism can actually be biomarkers of importance for clinical validation. So thank you again for do the work you do, because you have no idea how difficult it is to access samples with a really amazing phenotype. So I was thrilled that Marion gave me the opportunity to come to France and work with your samples to inquire the relationship between, oops, I passed on too fast, to inquire the relationship between lactate, which we're calling this metabolic shift in these patients, which, as you can see, can be influenced for many things. It can be influenced by the genetics on your patient, infection levels, environmental changes. And we're asking ourselves, is this changing leading to a metabolic shift in the patients, increasing the inefficient cycle, the lactate production, and decreasing the efficiency of producing ATP in your cells? Can this be related to metabolic syndrome and cardiovascular vulnerability? So we have had the opportunity to work with Fundación Fundamental samples as our testing cohort and replicate that into the iGive cohort. Again, the paper is published, and you all have been so helpful to get this paper to a point where we published in a beautiful journal and in a very nice publication. So in summary, what we saw is that your patients with metabolic syndrome in the cohort of Fundamental had a plasma lactate that was elevated in compared to those with no metabolic syndrome. The levels of mitochondria circulating DNA, we did not find it, despite we actually thought we would, but our hypothesis was not correct. We did not find in that metabolic syndrome was leading to increased levels of circulating mitochondrial DNA. When we start asking us, can we actually segregate the patients and understand their metabolic profile on this? So what we did, we take patients that were in a higher portion of lactate and also had the prevalence of metabolic syndrome. And these patients, as you can see, they have an accumulation of alpha ketoglutarate, which is highly important for formation of glutamate in your body, and an accumulation of citric acid. Citric acid in mitochondria triggers the synthesis of lipids, and one of them being cholesterol. And when you see the patients that had normal levels of lactate and had absolutely no metabolic syndrome, their levels of profile are completely different of them. This is informative, and this is actually can help us or even to target treatments for them. Because patients with citric acid actual elevation, there are ways that you can treat that and restore back the metabolism in these patients. So then we went back and asked for the iGive cohort. Is the patients with acute bipolar disorder or acute schizophrenia had even higher levels of lactate compared to the cohort from the Fondation Fondamentale, the FACE-PD? And the answer is yes, they were even higher than that. And all the three of them are lower than the controls. The accuracy that we could find, the better one was actually for the sensitivity was for the schizophrenia compared to actually bipolar disorder is stable versus acute. So now I'm going to shift gears and I'm going to pause for a second if you have any question, because I'm going to shift gears to a little bit a different topic, which is brain organoids. I don't see any hand up or anyone unmuting, so I'm going to continue. So we start modeling the disease. So many years ago, this was a dream and a pipeline that we had. Can we access brain cells of the patients? Can we actually measure the same thing in the periphery and try to validate it in the brain? A few years ago, back in 2014, this started being possible with the availability of creating iPSC cells, induced pluripotent stem cells. So basically what we do, we take the blood of the patients, collect the white blood cells, and teach these white blood cells to go backwards in the first cell of your body called stem cell. And then we teach that stem cell to now grow back and become a cell of our interest. In this case, is to become a neuronal cells. Brain organoid sounds fancy name, but in truth, what they are is actually a sphere, a blob, with a lot of the cells that represented your brain cells. When we create cortical organoids, I mean frontal organoids, you actually see like a presence of neurons and astrocytes and oligodendrocytes in these cells. And they all represent, and very importantly, the genetic that carries by your patient. So you actually have in a dish something that are neuronal cells that represent the genetics of the patient in question. So that's what we start doing in the lab, and of course, inquiring more questions. So when we create these organoids, we can ask ourselves, do they have less cells, the organoids from patients with a bipolar disorder? And the answer is yes, they actually do have less cells. They have less efficiency production of ATP. And interestingly, we found that higher levels of glucose 6-phosphatase, a metabolite in these organoids. If you look with me for a second in this a little bit complex graph turned down, but this is the breakdown to glucose to pyruvate. Glucose, the first step is to get into the formation of glucose 6-phosphatase. If glucose 6-phosphatase is accumulating in here, it means is decreasing, in here, is meaning that this process is actually going to be affected by the turnover of this process. Something is blocking this to go forward. So what people have actually identified, not from my lab, but from others, is that the hexokinase is downregulated in these brain organoids, actually making less efficient the turnover of glucose to glucose 6-phosphatase. So then we ask our questions, is the mitochondria functional in these brains? Is the mitochondria actually having potential of mitochondrial membrane potential, which is extremely important because it means that they are firing, they are actually active. And when we see here, the patients with bipolar disorder have exactly the same, walk through the green graph now, exactly the same mitochondria levels as the controls. But the mitochondria in the patients with bipolar are less functional. They are less polarized, which means that they can't really fire properly as it would be firing in others. So the next question is, can bipolar disorder cerebral organoids show any aberrant electrophysiology activity? So here is just to show you that the organoids have the presence of GFAP with markers as astrocytes, and also MAP2, which mark the neurons. When we did this electrophysiology at 6 months after this organoid has been growing in the dish, we see that patients with bipolar disorder at the baseline of the have more frequent spikes. They have less amplitude, which was not significant, but they also have an average of duration that is lower. So what you see is basically they are firing all the time. They are out of sync and rhythm. The difference between controls and bipolar disorder is quite striking. So that led us to ask the next question. Okay, now we understand that is some aberrant electrophysiology. We know this is connected with mitochondrial dysfunction because we saw that there is accumulations of different pathways and there is changes in the TCA cycle, ATP production. But that can be actually any explanation with inflammation that we all know that is present in these patients. So we start measuring what we explain for you, the CCF mtDNA. And you remember that we didn't see it in the fundamental cohort alterations at all in the blood from the CCF mtDNA. But now when we measure in the brain organoids, we actually see accumulation of this circulating cell-free mitochondrial DNA. The next, we ask the question about a pathway called the NLRP3 inflammasome. Fancy name, don't need to worry so much about it, but basically what this is a pathway in your body that is sensitive to production of reactive oxygen species and the presence of the circulating mitochondrial DNA that activates the formation of this complex called the NLRP3 inflammasome. And when activated, they cleave pro-cytokines, leading to producing mature cytokines, and one of them being IL-1 beta, leading to release now inflammatory cytokines. So again, we went back, and these are organoids now at 9 months point, where we have beautiful astrocytes forming in these neurons by the presence of the GFAP. And this is really important because this process happens in astrocytes or microglia, does not happen in the neurons. So what do we see? This graph is a bit complex to understand, but I hope I can walk you through. So basically, if you look by the blue ones, when you have this marker that we measure to see the activation of this called ACSPECs, we don't have any change in the baseline. But when we give a treatment to these organoids and we say, hey, let me activate inflammation on you, let me get inflammation to this brain, look what happened to the patients with bipolar disorder. They are 275% more sensitive to inflammatory insults. So basically what this is saying is that patients with bipolar disorder have this NLRP3 inflammasome like a radar, is on demand all the time. Anything that goes wrong in the body that produces mitochondrial dysfunction is gonna activate it much more sensitively in these patients. So we ask ourselves, can we block this? And that's what we did. We gave them a compound that we created in our lab. There is a mix of several berries that have high anti-inflammatory processes. And we saw, again, we tested against a very common leading compound in the industry, which was able to block this activation of NLRP3 inflammasome. But also our compound was very sensitive actually to block this activation. So that's very interesting for us because it's a promising venue for possibly treating the bipolar disorder circulating inflammation in these patients. So I end with a beautiful quote from Jan that I leave you to read in this slide. But this is the big picture pathway. What do we see here? Patients with bipolar disorder have an accumulation of glucose. We all know the prevalence of insulin resistance. Insulin resistance lead to change the production of reactive oxygen species, putting the mitochondria already in alert, in a sensing mode that is saying something is wrong with this. So I'm going to try to now regulate myself, but this leads to decrease the capacity of doing proper oxidative phosphorylation that leads to proper energy production, accumulating processes like free fatty acids and lactate that will continue increase the levels of reactive oxygen species. Again, putting this mitochondria in more and more sense. Unless we find ways of identify what pathway is from this chain is changed in our patients and effectively stop this chain of reactive oxygen species in our patients, we won't be able to properly treat it and restore their metabolic health. Here is just to show you the website of MitoCHY. So it's www.mitochy.ca. As I said in the beginning, please join. It's all for free and it's great resources if you are interested in this field. Thank you very much for listening to me, and I'm now open for questions.
Thank you very much, Anna. That was really, as usual, amazing. And I think it's so important for our patients. So I can't see any questions, but I have two questions while our colleagues are thinking about what they could ask you. Do you have any hypothesis on how the abnormal energy metabolism can explain manic episode and depressive episode?
Yeah, it's a great question, Mariana. And I think that's the gold mine question that everybody wants to be able to answer. My hypothesis is that mitochondria is a sensor in your body. And when the mitochondria is actually getting into your normally patients with prodromal mania, they actually have already an alteration on the energy demands in the body. They're going to speak faster, they walk more, they're more sexual drive, they having racing thoughts, and all of that requires tons of energy of your mitochondria to keep that up. And I think that is mitochondria just sensing to trying to compensate everything that it can. But it gets to a point where it breaks down. And I think that's the point when it breaks down that you see the full mania episode, which is a bit controversial what we thought. We thought that the majority of the time it's just like mitochondria putting a lot of ATP, and that's what is related to the mania episode. And then it goes down to low levels of ATP. I don't think it's that simple. I think it's really that mitochondria being this little thing that is sensing everything in your environment and just really trying to keep you alive and compensate for everything. But that compensation going to have severe consequences to the any organ on your body that is actually suffering from. In this case, the brain, which will lead to then full-blown episodes. And the compensation can now cycle back. And mitochondria are going to try to compensate because you gave antipsychotics to the patient, you monitor them back. So mitochondria is now trying to slow down and try to repair the damage that has been done, the inflammation that has been caused, the alterations on the cells death that leads through the mitochondria. So mitochondria is going to enter in repair mode and going to try now to repair everything that is there. But that costs energy too, and a lot of it. And then you're not going to have energy enough to actually lead to neurotransmission. And then maybe is when you see the cycle back into depression. Very difficult to prove that, very difficult to actually measure that. I don't think we have models yet that can answer that question biochemically properly.
Okay, but do you think that we could think of depression as being the cost to be paid for manic episode?
I think so. I would agree with that statement.
Then why are there patients who are just having manic episode? And could anything you found on those patients? Because then they wouldn't have this depressive
Yeah,
cost for the
I have not studied this type of population, and I'm unaware of anyone have done. So if you all have access to samples like that, that's a question to ask.
Julia has a question, but I'm sure we have in the database some patients with unipolar mania. And that would be worth studying because, if this is the cause of depression, then we should be able to find something different. Ophelia.
Yes, thank you, Hannah, for this wonderful presentation. I was wondering if you think that all of that is specific to bipolar disorders, or if it's also something that we can find in schizophrenic patients, for example.
Yeah, no, great question, Ophelia. It's great seeing you. No, I don't think it is a specific of bipolar disorder. I think alterations in mitochondrium are a trait, a marker that is a metabolic alterations in your body that is leading to cost demand of your brain to repair and to stay alive and to try to get itself in control again. So I don't think this will be specific of bipolar disorder. I do think schizophrenia you will see very similar or perhaps even more accentuated trends in these patients.
Thank you.
You're welcome.
So maybe another question, Anna, can you comment on the different treatments that are being developed for the moment and published, like ketogenic diets, the launched by Michael Burke in Australia, and explain maybe to our colleagues what is the hypothesis behind, for example, ketogenic diet?
Yeah, thank you for that question. I think so. I'm going to start by sharing again my screen just so you can see this slide as I speak through. Okay, so let me start by the ketogenic diet. The ketogenic diet has currently 12 clinical trials around the world, not only in bipolar disorder, but clinical trials in schizophrenia and also in ADHD. And the hypothesis of ketogenic diet is to decrease the amount of circulating glucose in your body and increase the amount of ketones. Ketones are another source of energy. They bypass this entire pathway. They don't need to do any of this. So they relieve your body of actually any time entering insulin resistance, lactate production, or increasing free fatty acid productions. The ketones enter directly in the TCA cycle around this part on the alpha ketoglutarate acid, leading to a speed up. Like it's like if you hit a speed bump in the road, to speed up your energy production, relieving the body to have to use a lot of the pathways that actually are damaged in these patients. So is hope for the ketogenic diet is a way of restoring the mitochondria pathways, restoring oxidative phosphorylation, and increasing the availability of ATP in your body. The first thing you talk to patients that have effectively gone to this treatment, the first thing they say, I don't feel tired anymore. As the first thing that you entered on the ketogenic diet is a relief of brain fog and the fatigue is decreased. So I think is a great potential of treatment for these patients, but it's too early to say and how to effectively do. Some of the other clinical trials have tried to use antioxidants. So Michael Burke, for example, created an antioxidant cocktail which take all these ROSs that are around and try to remove them of the equation. So if you're removing reactive oxygen species of the equation, again, you're giving time to mitochondria to breathe, to mitochondria to restore itself, to actually try to get better. So that was effective on the first trial for patients with bipolar depression. But in the second trial they did, they identified that actually in the long term, it led to cause mania more often in the patients. Not sure how and why, but that was a finding that they are trying to investigate now how to overcome. Then there are many other type of molecules like that you can regulate specific parts of this TCA cycle, so the metabolites. So I believe Michael Burke has a new molecule that he's trying to work on regulation of the malate on this pathway. So it kind of, again, it's the same thing that we just said for the ketogenic diet. It's trying to boost this and relieving this whole part of glycolysis to improve ATP production. Very similar thought of the ketogenic diet, but now via a molecule and not a diet. Another trials that have been tried is to actually restore mitochondrial function by increasing biogenesis. Biogenesis is a big term, but basically just means that mitochondria divide itself and it can actually get healthier because it's doing more fusion and divided and creating more mitochondria in your cells. So Andrew Nierenberg has tried that with bezafibrate, which is a molecule that increases mitochondrial biogenesis. But I think he never ended the trial because the bezafibrate unfortunately become not available in the United States anymore, which was such a shame because the 6 patients that we did it the first time was unbelievable to see the trend, how they would improve, but we never were able to conclude that trial. So yeah, that is to answer your questions. There are several treatments. Ketogenic diet right now being the most promising one that is available there. It's not the easiest diet to follow, but I have tried myself, and I love vegetables and meat. So I'm Brazilian from the south of Brazil. Give me a piece of red meat, I'm there and eating all the time. So for me, it was not difficult to, but when I was in France, I love my croissants, so that was hard to follow when I there. So it's hard to make sure that patients can follow that. So I do think we need strategies there. First, to do identification. What are the metabolic types of your patients, and we can use small molecules that can probably restore. And then when the patient is better, teach them to get in a diet that is more plausible for them to do. I think it would be very hard to conceptualize it that a patient in a manic episode can change their diet from day to night, right? So I think we need more strategies of thinking. How can we actually treat patients that have metabolic alterations?
Well, thank you, Anna, for all these examples of treatments and clinical trials ongoing. Just so that we maximize the chances of success, what would be your best guess on how to identify the patients, for example, who are going to respond to ketogenic diet, which obviously is not for everybody?
Yeah, that measuring the mitochondrial metabolites, exactly what we did at Fundación Fundamental, the cohort to the paper that we published with you, that we had that graphic stratification that I showed you. If we can do that, measuring the mitochondrial metabolites, but we need to answer the question properly, you're going to need the longitudinal assessments. So then we can see if the patients are changing over time, and then we can find the biomarkers that are actually useful for you. Simple example. If a patient has an accumulation of citric acid in one of mitochondrial metabolites, you might want to consider in this patient lipoic acid, because lipoic acid is a substitute of citric acid. So we will, again, it's all about relieving the mitochondria and give space to the mitochondria to repair itself. So you're now saying to the mitochondria, you don't need to use that citric acid anymore. I gave you lipoic acid. So you're telling the mitochondria to not sense and triggers the activation of production of cholesterol in these patients, which is what citric acid does in your cells.
So we need personalized diagnosis as well as personalized treatment, if I follow you
Yes.
well.
Yes, that's what I kind of say. No, that is not it's not easy to do it. But we have done effectively in cardiovascular diseases and in cancer. So I don't think it's a big of a leap of faith to say that we should take it and we should try it to identify them. It's a simple mass spec and it doesn't cost very much and it's easy to do it and it's already done for mitochondrial rare diseases, is already clinically used. So that is clinical availability of these tests is around there. So we just need to continue replicating the data, and I think that's what we're doing with you and with Mark Fry in the United States, and really trying to showcase that this is not a one-time finding, because I think that's the biggest problem in science we have. We do it once and then we just don't do it again, and we don't replicate our data. And by not replicating our data, we don't convince people that that's a very important finding. Maybe we can just say, like, remember that for you to breathe, you need a mitochondria, you need a healthy mitochondria. So keep treating your mitochondria well. If anything, I say to everyone is like, think about your heart, what you do for your heart, what your cardiologist says, sleep well, do a good walk, exercise, eat well. That's what you do to keep your mitochondria healthy and happy, to keep you breathing.
Well, with these very wise ideas, maybe we to conclude if no one has any question. Anna, thank you so much
You're very welcome.
doing, and thank you for sharing all this information with us in such a clear way. And we look forward to the next collaboration. Thank you very much. Merci à tout le monde et à bientôt.